Alright, at the end of that 5-minute centrifuge spin now, the pelleted cells are being removed from the centrifuge, of course, back over to the CO2 cabinet, and you can see now there's a pellet of cells, hopefully, at the bottom of that tube. Remember, it's a small pellet, so we've got to carefully take off that supernatant without losing any of that precious product at the bottom. So cap being removed from the centrifuge tube of the cells. That supernatant of trypsin now being removed carefully without disturbing the pellet at the bottom. That trypsin into the waste. And now the freezing mixture is going to be added to this pellet of cells. Now remember, the freezing mixture is a mixture of your normal medium and usually with an additional amount of fetal calf serum added, depending on the cell type and the procedure, and importantly with a certain percentage of dimethyl sulfoxide added to that freezing mixture. Now remember, the purpose of the DMSO is to prevent the formation of ice crystals as this aqueous mixture of cells and media is going to be frozen. So here's the addition now of 5 mils of that mixture of media, fetal calf serum and dimethyl sulfoxide, and being titurated up and down now through a sterile 5 mil pipetta tip. Now that tituration process, as I've mentioned before, is to break up that clump of cells, get those cells separate so that we have an even, suspension now of those detached cells ready for the freezing step. So we'll see another 5 mil addition here of the freezing mixture to that tube to make a roughly 10 mil total. Into the tube now. And again, mixed up with that tituration process, plunging it up and down through the pipetta tip to ensure the mixture is even and those cells are roughly evenly distributed through that freezing mixture. Now the next step is to aliquot that freezing mixture into the cryotubules, small little robust tubules, and to be stored into the Mr. Frosty, which you can just see in the background there, just between Hannah's hands as that view. Now again, a sterile 1 mil pipetta tip. Here's the Mr. Frosty, the lid's being taken off in preparation, and here now are the cryotubules. So what you're seeing here is a pre-prepared set of the cryotubes. These are the small 1 mil tubes, robust that they'll tolerate that freezing process, and with a conical bottom so that you can easily recapture the cells once they're thawed to be recultured. So here now, again, dispensing that mixture of freezing mixture and your cells into the cryotubules, tubes, and now into that Mr. Frosty. Now again, for each aliquot taken from that mixture of cells, the pipetta should be titurated again, because remember, those cells will settle to the bottom. So ensure that your good procedure is mixing, keeping that tube of cells resuspended for dispensing into the cryotubes. Now these cryotubes are going to be stored at -80 in our -80 freezer. But of course in many workplaces they would use liquid nitrogen. But for our teaching purposes, -80 is quite an adequate storage. Now the Mr. Frosty itself is a plastic robust container that does contain an isopropanol alcohol solution down the bottom here, which will cause rapid freezing and cold or heat cold transfer into those cryotubes in preparation here for transport over to the -80 freezer. So there's our now small 1 ml aliquots of that freezing mixture and cells ready for storage. And just remember, the whole purpose of this is to keep your cell cultures frozen for when they are next needed. Okay, so it's a preparation, if you like, stocks of cells ready to be rethawed, recultured and grown up again for whatever purposes are required. And I think that's about it. Thank you very much, Hannah. Well done. And yeah, we'll look forward to hear the cryo thawing process which follows on from this.